The Immortal Life of Henrietta Lacks, which is a bestseller. It's an absolutely amazing book for any of us who are in the field, especially non-scientists, to understand how a bunch of human cells could create a billion-dollar industry, and the profound ethical questions that are raised in a really compelling read. I certainly recommend The Immortal Life of Henrietta Lacks. As this panel sets up, our esteemed moderator at Goldman will introduce himself and his panel. And I want you to know that immediately after this panel, from 10:30 to 11, we have a break at level 4, this level in the foyer, where we're going to have refreshments for you. And then we will resume at 11 o'clock with a scientific keynote address from Dr. Sean Morrison of the University of Michigan, followed by a keynote address jointly with Dr. Eva Feldman of the University of Michigan and A. Alfred Taubman, following with some additional remarks, if I'm not already exhausted all my remarks, and then we'll have our expert luncheons. Thank you.
Good morning. I'm Ed Goldman, another one of those lawyers who have strayed into medicine somehow or another. I'm from the University of Michigan. I've been active in stem cells for a number of years now. I helped with the November 2008 election that legalized creation of stem cell lines in Michigan. I work with the Department of Obstetrics and Gynecology and the Center for Stem Cell Research at the university. I sit on the university's Embryonic Stem Cell Committee. I chair a committee on ethical uses of stem cells at the university, and I know just enough medicine to be actively dangerous and misinformed. I have really a distinct honor today of introducing our panel. We are going to be discussing the proper use of tissue that would otherwise be discarded. How can that tissue be used for research? What consent ought we to require? What information ought a patient to have? What have we learned from the Henrietta Lacks story of tissue being removed for clinical purposes and then being used to create a cell line without consent from the patient or her family? What about removal of tissue with consent for a purpose and then using the tissue for other purposes? Some of you may have seen the settlement in the Havasupai Indian case in Arizona. The Havasupai, who live at the bottom of the Grand Canyon, thought that the Arizona State University researchers were removing their tissue to study one specific issue, diabetes, but in fact tissue was then, or DNA was then shared with other researchers who used it for schizophrenia studies and for migration pattern studies without consent or knowledge of the tribe. So what we're going to look at today is how can the past help inform the future of embryonic stem cell research? What do subjects want to know? What do they deserve to know? What ought they to know? How should they be told? We have three distinguished panelists. They each have 15 minutes to tell you everything they know. That, of course, is not possible. They're going to do their best to give you their current information in thinking about these critical issues. I'm going to introduce all three of them and then get out of their way and let them use their time most productively. Looking at their biographies, if I were to fully introduce them, there would be no time left for the panel, so I am just going to hit some of the high points. Dr. Jeremy Sugarman is an MD, an MPH, an MA, and an overachiever, and the Harvey Meyerhoff Professor of Bioethics and Medicine. He's Deputy Director for Medicine at the Berman Institute of Bioethics at Johns Hopkins. Johns Hopkins is, of course, where the Henrietta Lacks story began. His work concentrates on informed consent, research ethics, global health, and the ethical issues associated with emerging technologies. He's the author of almost 200 publications in peer-reviewed journals. He's an associate editor of clinical trials. He's on editorial boards of several institutions, and he serves on the scientific and Research Advisory Board for the Canadian Blood Service. He's a member of the Maryland Stem Cell Research Commission and co-chair of Hopkins Institutional Stem Cell Research Oversight Committee, and a fellow of the American Association for the Advancement of Science, American College of Physicians, and the Hastings Center, the oldest bioethics think tank in America. Dr. Laurie Zoloth is director of the Center for Bioethics, Science and Society at Northwestern, the Feinberg School of Medicine, where she teaches medical humanities and bioethics, and is also a professor of religious and Jewish studies at Northwestern's Weinberg College of Arts and Sciences. She directs Northwestern's Brady Program in Ethics and Civil Life. And Laurie has done things like help establish basic organizations like the American Society for Bioethics and Humanities, receiving its Distinguished Service Award in 2007. She's also helped establish the Society for Jewish Ethics. One of the fascinating things in her biography is that she has been a member of the NASA National Advisory Council, the nation's highest civil advisory board for NASA, and received their National Public Service Award in 2005. She is on the executive committee of the International Society for Stem Cell Research. She founded the Howard Hughes Medical Institute's Bioethics Advisory Board, been on founding national boards of the International Society for Stem Cell Research, Society for Scriptural Reasoning, and NASA's International Planetary Protection Advisory Committee. She's had numerous honors. I'll just mention one or two. In 2005, she was honored as a Graduate Theological Union's Alumni of the Year. In 2009, she received the McCormick Professorship, Northwestern's highest award for teaching. As long as you're in Michigan, Laurie, since it appears that you have no ability to sleep and you work all the time, we do have a sleep lab, and we'd be pleased to help you with this problem. Chris Hempel has 6-year-old identical twin daughters, Addi and Cassi. They are afflicted with an ultra-rare fatal cholesterol storage disorder called Niemann-Pick type C disease. This condition is often referred to as childhood Alzheimer's. And while it is an ultra-rare disease, everybody in the world is born with a Niemann-Pick C gene, which regulates cholesterol metabolism at the cellular level. So what scientists learned about Chris's children might someday help millions of people with cholesterol-related diseases. Since Chris's children received the diagnosis in October of 2007, she has worked tirelessly with doctors and researchers around the world to search for a life-saving treatment for her daughters. And in May 2010, in association with Children's Hospital in Oakland, she was able to get the FDA to designate an orphan drug for a non-toxic sugar compound, which turns out actually to be the active ingredient found in Procter & Gamble's Febreze fabric refreshener. Less than 2 weeks ago, Chris and her doctors received FDA clearance to begin the world's first cytodextrin administration directly into the brains of her twins in an attempt to stop the neurological progression of NPC. Treatments are expected to start in a few weeks at Children's Hospital in Oakland. So you're going to be hearing very current information from all of our speakers today. Over the past 18 months, Chris has been working closely with the leading MIT researcher at the Whitehead Institute to look at induced pluripotent cell lines from her daughters and differentiating them into neural progenitors. Prior to becoming a full-time medical advocate for her children, Chris worked in corporate communications at Netscape and co-founded Spark PR, one of the leading technological public relations companies. You can learn more about her story and the Rare Disease Initiative at her website. Easy to remember, it's her twins' names, AddiandCassi.com. Let me turn the program over to Jeremy.
I used to place to get up and talk, or do we sit here?
Thanks so much, and good morning. I wanted to ask how many people had read this book, The Immortal Life of Henrietta Lacks, and I can only see the front 2 or 3 rows, so the rest of you don't even have to show. No, seriously, how many people? Small portion. Okay, well, for those of you who haven't heard of this book, where's the slide advancer, actually? That's not the question. Do we have one? Oh, there we go. There's the cover of the book. It's been on the New York Times bestseller for a good period of time, and it chronicles the life of Henrietta Lacks, and you can look at those first 2 initials, H-E-L-A, of her last name, and suddenly for science that takes on new meanings, the HeLa cell. Henrietta Lacks was the person from whom cancer tissue was collected and made these famous cell lines that have been unbelievably important in all sorts of science, ranging from across the life sciences, and it's taught us everything from cell culture, work on polio vaccines, HPV, and the like. You cannot have done a degree in biology or anything related to it and not have used HeLa cells or learned from HeLa cells. The part is that this story began well over a half a century ago when Henrietta Lacks came to where I work, the Johns Hopkins Hospital, and there the older building in the front is the hospital in which the cells were collected when she came into that hospital. And the glitch is that these cells were taken without consent, as was the habit or the practice of the time. Over 50 years ago, materials were taken from patients or persons without permission or consent. This was not in itself surprising, but it's the story, it's the legacy of these cells that have gone on to capture the popular imagination and is beginning to teach us things about the ethics of research then and today. Not to mention that Henrietta Lacks' entire family is now aware of this story of the HeLa cells that have gone forward. There are questions about commercialization and use and a variety of concerns that are raised. Just this past Saturday, we celebrated at Hopkins the first Henrietta Lacks Memorial Lectureship featuring Rebecca Skloot, the author of this novel, which was a community and Hopkins community joint lectureship that filled our largest auditorium at Johns Hopkins. We were oversubscribed. And so in the East Baltimore community, which usually doesn't come past the doors of our hospital unless they're sick or they work there, people engaged in a conversation about HeLa cells and the perceptions and misperceptions about what science had to offer then and today. The stories aren't all good. Science always doesn't come off looking great, and it gives us an important obligation to get these things right the best we can at the times we're doing them. In addition to that, beginning this inaugural lectureship to honor her contribution, there is a scholarship fund that's been established for talented high school youth across our street at Dunbar High School, usually referred to as a place with the great basketball players that come out of Dunbar, but here we're going to privilege science, as well as a yearly award to the community organization that engages in research. So I just had to do that brief diversion based on my sampling there. Okay, next slide. The only sort of scandal in the recent year wasn't related just to the HeLa cells or Henrietta Lacks. Ed referred in his introductory comments, this is a photo from the New York Times piece, when the Havasupai tribe settlement with Arizona State University was taking place earlier this year. And in that case, what happened is the researchers from ASU went to the Havasupai initially to gather biospecimens to be used for diabetes research. However, the researchers had these bank specimens and decided that they wanted to explore some other hypotheses, things related to things like schizophrenia, and also to the ancestral origins of the Havasupai, which played against their own origins, their own myths of where they came from. The Havasupai were angered over the unintended use of those tissues, and took action against Arizona State. This is an enormously important case because things that are in freezers or things that live, like HeLa cells, or the materials that the Havasupai gave for one purpose being used for other purposes, have direct implications for what we do. And finally, in the lower right-hand side, you see blood spots. And the blood spot issue is now playing out in several states about whether these blood spots, which are done for neonatal screening, under a public health imperative, can and ought to be used for research. And now there's a backlash against sort of those moves. So science is not taking place in the vacuum. The political science, as the governor said, is very much part of what we're doing. Next. So what I want to do, not intending to have given you that background, but now that I have, talk about how things have changed with respect to when samples were taken, say, over 50 years ago in the case of Henrietta Lacks to now for biobanking in general, and then in stem cell research in particular. I'm going to touch on the implications for the guidelines we use for the oversight of pluripotent stem cell research, talk about some challenges and opportunities in 4 areas. And I'm going to do that sort of quickly just to show you how these things that are occurring in the world around us are very much going to shape what we ought do in stem cell research. Next. Modern biobanking is very different from the days that Henrietta Lacks had her cells taken. Their proliferation of large-scale biobanks around the globe, their proposals for approaches to best practices, which include attention to the ethical aspects. It's no longer okay simply to take tissues and throw them in a freezer, give them to the person's lab next to you, and use them whichever way you want. One example is the new proposal for the NCI best practices. This is only one of many, many, many around the globe. But this one you can see explicit attention to issues of consent, ownership, and the like. Next. For ethics in biobanking, when you look across the globe and start thinking about what do we need to consider from a moral perspective when constructing a biobank, in addition to all the things you need to do to make sure that those materials stay tasty enough for science, are issues of benefit sharing. Who benefits? There's been a broad discourse among the Lacks family, attorneys, and the like about what benefits, if any, should the Lacks family have as a result of what happened with HeLa cells. Consent, clearly a part of what we do now, but how thorough should that consent be? What can we anticipate in the future? Privacy and access. One of the most compelling issues of the Lacks case is actually that inadvertently all the Lacks family was brought into this story of their now great-grandmother's cancer, her cervical cancer, and what that means, and the fact that her mother's cells or grandmother's cells or their aunt's cells are somehow alive in a way that doesn't comport necessarily with the way we think of things in science, but have very different meanings among some people. And finally, governing bodies. What kind of governance structure should we put into place in order to make sure that we're doing science in the right way? Next. Well, the background conditions for stem cell research, scientific excitement sound. Do I have to tell this crowd that? We're jazzed. This is the best thing. This is great. We love this stuff. It's going to do a lot of things. It may not do the things we're hoping it's going to do, but we're going to learn from stem cell research. We already are. The desire and access to investigational treatments abounds, especially for people with devastating disorders. Again, not a surprising statement, but that is what has made some of stem cell research be able to move forward, is that there are certain things that we cannot currently treat or cure, and people want access to those. John Kellerman, the man that Bernie mentioned earlier, who received the award from the organization last year, was a passionate colleague on the Maryland Stem Cell Commission with me. And he was also a sort of a pioneer because he received the first DBS, deep brain stimulation, for Parkinson's disease at Hopkins. And he got this thing, and there was a picture of him beforehand, unable to walk, unable to move, turning on his deep brain stimulator and being able to play lacrosse with his sons. Totally remarkable. Totally remarkable. He said to John, well, you've got this DBS, why stem cell research? And he said, okay. He started to turn this off. I turn off the switch, and I'm back. Do you know how to turn it back on? I said, no, it's not my kind of medicine. He said, well, it's easy enough to turn on, and I won't let that happen, but I want to be cured. I want something to cure me, and only regenerative medicine was the approach. The financial and moral stakes are high. The political science we just heard about was about state governments investing, industry investing. There's a lot of money being directed at this with hopes of financial success as well as scientific and medical success. The stakes are high. And there's an enormous trust in the scientific enterprise that we get this right. Next. Professional guidelines grew up. Obviously, human embryonic stem cell research has not been value-free. Starting with embryonic stem cell research and questions about the moral status of the embryo, but also all the state differences that existed around the country in the United States and the international differences that took place. For instance, it is a criminal offense in Germany to create a human embryonic stem cell by destroying an embryo. It is a criminal offense. So what if a German postdoc comes to the University of Michigan or Michigan State or Wayne State and learns how to create an embryonic stem cell line? When she goes back to Germany, she'd be thrown in jail for that? What's a mentor to do? What's a journal editor to do of a major scientific journal if that science is important? That's just one example. So in light of all these differences, next. The National Research Council and the Institutes of Medicine, next, and the International Society for Stem Cell Research, each crafted sets of oversight or professional guidelines because this research, because it was happening at the bench, fell through the cracks of our typical oversight systems that are used if we're doing research with animals or research with people. Next. So the recommendations were that we should apply sort of voluntarily oversight for human embryonic stem cell research. Under NAS guidelines, these were called escrows. It sort of correlated with the mortgage crisis, so I'm not sure why. Under the ISSCR, it was the Institutional Stem Cell, or SCRO, Stem Cell Research Oversight Committee, because the ISSCR took more broadly all forms of stem cell research. Next. The basic idea is that you'd have a committee that would deliberate about these uses in order to help negotiate these places where federal laws and state laws might be silent or might be operative, and also where there were international differences. Next. Recently, we've seen the NIH guidelines change after President Obama's rescission of President Bush's order, where there was an establishment of the Registry of Lines. There was a working group to explore which stem cells are eligible. There were paths for approval of when stem cell lines could be used because of debates like the HeLa cells. Where did these cells come from, and would that research be okay? These are not unimportant issues. And then they put in place also certain prohibitions. Next. So what are we left with, even if we have these oversight committees? Well, we have challenges and opportunities ahead, which the case of Henrietta Lacks and other cases like the Havasupai, the blood spot case, can inform us. About 4 areas: provenance, MTAs, which are material transfer agreements, those sort of forms that are used when scientists want to transfer things between labs and use them, induced pluripotent stem cell research or other human pluripotent stem cells, what's happened now that we are not relying solely on human embryonic stem cells, and finally clinical research. Next. So this graph is one that we proposed prior to the time when the NIH working group was established. And they say, what do we do when a researcher at Hopkins, or anywhere else for that matter, wants to use a particular human embryonic stem cell line? Is it okay? Now, several places opted for this idea that you would use a line because the federal government said it was okay to use the line. Ethics and politics are not necessarily the same thing. We've seen it change. Aristotle had separate books, had it right a while back, but ethics and politics are different. So we said, well, we're going to make it harder on ourselves. So if someone proposes a use, starting at the top, and go down the left side there with green, evidence indicates that the derivation was in accord with whatever guidelines existed at the time. Did you get consent? Did they prohibit anything? Did they understand the embryo would be destroyed and the like? You could permit this use. One case we had was a very famous set of lines, which were approved for use by the federal government, which were created, and when we looked at the provenance of those lines, we found that the donors of those embryos, discarded embryos, thought that they were giving those embryos off for research only on diabetes. Our scientists on the committee said, you can't use those for this proposed research because this proposed stem cell research has no bearing on diabetes. You can't use them. In contrast, another set of lines, which were not federally approved, which were an industry set of lines, we looked at, and they weren't approved because there wasn't language in the consent document that specifically mentioned certain stipulations, but it was clear from the context that they did, we allowed those use. So these are really tough deliberations that take place. Okay, next. I'm going to speed up. The most recent debates about this are the RGI lines that have come out, in which these are the largest set of disease-based lines that had been existing, and yet the working group at the NIH disallowed their use because there was exculpatory language within the consent documents that people signed under the piece that talked about patents and ownership. And the question was, did this somehow undermine the patient's interest in that research that gave this over, or the subjects? And it's an incredibly important set of questions that we're wrestling with now. Will we allow this to go forward even though the federal government won't? Next. Expanding the purview behind embryonic stem cells. Does this matter? Well, scientific developments have brought lots of excitement. iPS cells, induced pluripotent stem cells. Why does it matter? If the embryo questions are over, why can't we just do whatever kind of research we want to do? Well, this is the example we think of first. If you take a skin cell, it's just a skin cell, you'll leave several on your brochures and things, and we could collect them. We just take your skin cells and we make it into a stem cell. Fantastic science. That is very interesting. It's really interesting. It's really important. If you're going to say it's going to go into any kind of stem cell, any cell, because it's pluripotent, well, one of those kinds of cells is sperm and another kind of those cells is eggs. They look like sperm and they look like eggs. How do you prove they're sperm and eggs? Basic science. You put them together. Well, you've just created life if it works. So you go from this situation where you take a skin cell and you make a person. We're back full circle to where we were with the human embryonic stem cell debate. That's one example. The HeLa cells and the Henrietta Lacks case tells us that people care about how their cells are used, and pluripotency, not to mention just being immortalized, but pluripotency might raise some critical questions for a society that supports this research. Next. So now our committee has been changed from the escrow to the ISCRO. So we now do ethical oversight of all human embryonic stem cell research and or somatic cell nuclear transfer. But we also do oversight at Hopkins of other human pluripotent stem cell research if it involves putting them into people, putting them into the central nervous system of non-human primates, introducing them into non-human animals where there's a significant possibility of the cells giving rise to gametes or the creation of gametes or embryos. Federal laws are quiet on these issues. We are applying oversight at Hopkins, not just because of Henrietta Lacks and the HeLa cells, but because we started this approach and really believe that this area raises important moral questions. Next. Moving forward, our questions are going to be, what do we do about approval and consent for collecting biological materials anticipating they'll be used for producing pluripotent stem cells? What does our IRB, our Institutional Review Board, need to do, not just to say, oh, you can take it and leave it, but going forward, what additional information needs to be there? And what do we do with these treasure troves we have of Hopkins, of biobanks that have tons of material that people would like to use to create pluripotent stem cells? We're working through those. Next. Material transfer agreements, this next area, it's been sort of ignored. It's been something that, one person sits in a small office in most medical centers and in labs and processes agreements so that when the FedEx guys or the UPS guys will come over to pick up the materials, that they have a form that says you can use this for something. What we've learned from the Lax case and what we've learned from deliberations about human pluripotent stem cell research is that those uses matter. Sometimes the uses, when you send something from our university to another place, what's going to happen there doesn't give people a carte blanche to use them wherever they can. So what we've done now is we're just starting to roll this out, is introducing a checklist for researchers to look at relevant moral distinctions. I'll be happy to answer more questions about these later. Next. Finally, I'm going to close down on this area. The excitement that we have is really going to be related to clinical applications of stem cell-based interventions. This is an emerging area, not directly from sort of the Henrietta Lacks story, but one in which we really need to get ourselves together about what's going to happen as we move forward to clinical trials as we're doing, and as we negotiate the various different pathways to where stem cell interventions are being done. They're clearly a set of inappropriate uses. You just simply have to go to the web to see a lot of those inappropriate uses. Physicians around the globe have a fiduciary responsibility to their patients to take care of them and not use things that would be harmful, and to be looking for things that are helpful, and we need to come up with responsible ways of innovation. In the interest of time, I'm going to skip what I was going to talk to about that super exciting area. We get to that step. Fortunately, we have structures in place with respect to clinical research. It's with innovation and that area that we're starting to see new guidelines, and we can talk about those later. So thank you very much for your attention.
Okay, I need some slides so I know what to tell you. Okay, so I'm Laurie Zoloth. I'm from Northwestern University, and I'm going to have to say next each time. Right? Yes, next. I'm going to try to do something new. My colleague Jeremy Sugarman has given his usual excellent talk about the standard way that bioethics works to think about human embryonic stem cell research and to figure out what to do, giving the standard ethical norms. I'm going to try something new. In honor of Michigan's having a new line, I'm going to have a new line in bioethics, and I'm going to try to prove to you that a new line is needed just at this moment of crisis, because the situation that Jeremy described, that the book actually sets up for us, has some inherent problems. I want to challenge the language about the giving of the self in medical research, and give you a perspective not only from ethics but from religious studies, which is my other discipline. Next. I'm going to call this Fidelity, and I'm going to say, as we begin, medical research on human diseases requires participation from human subjects. And if it is shut down because of our inability to give freely, in a sense, the research enterprise really can't go forward. So people who are donating, exchanging body parts, body tissues, or having research done on them actually have an extraordinary responsibility and power in the relationship. It is in fact true that if Henrietta Lacks had been asked about her tumor cancer cells and had for some reason said, no, don't use them, medical research as we know it would be substantially different. And in fact, probably many of us wouldn't even be alive today because of the next story I'm going to tell you, because of polio. All codes about research derive their power from two different stories, two different narratives. First is this notion that there's a long history of medical ethics that for hundreds of years assumed that medicine had an inherent norm. There was an inherent professional norm that protected patients and subjects. And we allowed, for hundreds of years, doctors to do almost anything to human bodies to get the answers they need. After an initial skirmish in early modernity about whether you could use the bodies of the dead, right, which was initially resisted, once people had decided you could use the bodies of the dead, increasingly we decided we could use the bodies of the living. Why? Because doctors had an inherent sense that they needed to protect their patients and that they should do the research within a code of particular honor. And when this failed, the second narrative became the powerful narrative of bioethics, which is you can't trust doctors. They don't do a good job. And yesterday's headlines about the Guatemalan research on Guatemalan subjects for injecting them with syphilis into their spinal cords and then seeing if penicillin worked was the end of that sort of narrative. Well, I hope it's the end of that sort of narrative. With a long history that cuts against the story that doctors are good and that medicine itself shapes them in good and decent directions, because there's this other story that doesn't, that ends in Nuremberg and Nazi doctors and terrible experimentation on helpless human subjects. And this contradiction between what our vision was and our aspiration was and what had happened in the Nazi experimentation on human subjects resulted in the Nuremberg codes and a series of other codes that then govern research. Next. There was a shift of our attention from the doctor's concerns to the subject's concerns. The first idea, the doctors are good and they're shaped by their profession to do good, is based on the philosophical principle of beneficence. It relies on the doctor to do good, or at least to do no harm or do nothing at all. The second idea, the second story, is based on the power of autonomy, another principle in bioethics. It relies on the subject's own understanding of possible consequences. Next. But you can see that both of these things have problems, and they're both challenged by the kind of new research paradigms that Jeremy just described to you. Doctors and scientists have a sort of a sorry history of being tempted, of having conflicts of interest, and in some of these new revelations, cynical abuse in the name of fame or pride. We see that all too often. And subjects, as in the Henrietta Lacks story, may not have the entire story. They may come to feel later that doctors are getting wealthy, and then why not them? And the transformations of tissue may seem to be different in a culture that understands DNA as the self. I will wager to you that this notion that your tissue is you, that Henrietta is actually not dead but is immortal, is based on this idea that the cell, the DNA, has a life beyond the actual life of the person, which I'm going to suggest to you is an essential fallacy. Next. Autonomy, I believe, has its limits. Autonomy and freedom act as brakes on the power of medicine and the state. And in the Nazi experiments, we relied only on the frail but important power of liberal theory to create an autonomous choosing self that could say no to oppression, no to tyranny, no to domination, no even to doctors, and no even to the state. This sort of no, this sort of autonomy is tremendously useful in business or contract negotiations between strangers where limits must be firm, where the no must be firm. It's critical if the body is being used as the site of testing. But what if the tissue that's being used, what if the discussion is not even a part of the body, and in fact a piece of the body that everyone wants not to be in the body, as in a tumor cell? Next. The revelation about the fate of the family of Henrietta Lacks, whose tumor cells were used as the basis of most cell cultures, reveals not the abuse of the subject, I would warrant, but instead a very familiar narrative of race and class in America. If Henrietta Lacks had been, and she could have been because they took tumor cells from everyone who came into Hopkins, if she had been an upper middle class, white privileged woman, and her descendants had had lives of grace and access to health care, we wouldn't have the book, which makes you really wonder if the harms done had to do with the race and class narrative or had to do with the human subjects narratives. The idea of making tissue anonymous was seen, and de-identifying the tissue is a powerful idea within medicine that we don't want anyone to know. And that whole notion that you didn't say the cells of Henrietta Lacks and here they are, but instead you say HELA cells, was done to all patients in the name, by the way, of the highest possible standard of medical ethics at the time, to de-identify. And notice how that cuts against this idea of credit or reward for participation in research, which might be a new standard. Next. The case presents an important ethical question. Why? Because we all understand, and we all agree that instrumentally using the body of another can harm the body of the other. The gap between the situation of the family of Mrs. Lacks and the researchers who use her cells is quite apparent, and much is made in the book, in fact, of that tremendous poverty that the Lacks family struggles with, and what happened to them when their mother died. But are these the lessons that we're supposed to learn from this exchange? Especially for human stem cell research, which, quite frankly, colleagues, cannot proceed without this generous gift of tissue to researchers. Next. I'm going to go back and compare another case. In 1947, the case of Jimmy Sarkett. Now, who is Jimmy Sarkett? He was a poor white kid, a little boy, from a farm family. He had never left his town, on Daisytown, Pennsylvania. He had never left the farm, and he got a horrible case of virulent polio and was taken to Municipal Hospital in Pittsburgh, and he was left there in the children's ward because in those days parents couldn't visit, polio highly contagious. And then after he recovered from his initial symptoms, his initial illness, he was sent to the Permanent Rehabilitation Center, the D.T. Watson Rehabilitation Center outside of Pittsburgh, along with most of the children who had polio in 1947, 1945 to 1950. They lived forever at the D.T. Watson Rehabilitation Center because they couldn't breathe, they couldn't walk, and they needed complete custodial care. It was paid for by private donations. As he left the Metropolitan Hospital, the nurse drew a sample of Jimmy Sarkett's blood, mislabeled it Sukkot blood, sent to Jonas Salk, and this became the standard type 3 polio virus that was used for all subsequent research. Next slide. Now, in 1951, here we have our case of Henrietta Lacks. Notice the coincidence of time. It's in Johns Hopkins, but it could have happened anywhere, I would argue. She, too, from a poor but urban black family. Tissue here taken from a tumor that was going to be discarded anyway. Standard procedure to do so for all cancer patients. Treated the same, white women, black women, men, women, from all people with cancer. The cancer was sampled, and the HeLa cells were remarkable. They were used to test and grow, next slide, polio vaccine. And in fact, Jonas Salk used HeLa cells to test and to grow this Jimmy Sarkett's virus in those cells to create the vaccine that was then given to test subjects all over the United States, and including me. I was an early polio vaccine test subject, I might add. Conflict of interest, what can I say? Now, Jonas Salk, what's the difference between why Jimmy Sarkett doesn't have a book about him and why we don't know anything about his descendants? Right? And we do have deep concerns, and I too had the author of the book who gave a wonderful talk and came to Northwestern, overflowing crowd. What's the difference? Well, first of all, Jonas Salk made no money off a polio vaccine. Right? He did not patent the polio vaccine, famously said, you might as well patent the sun. And the Marsh of Dimes, who funded the research, did not expect or promise profit to the people who had invested in it. There was no circulation of marketplace or tax-saving promises like in California in the public understanding of polio. Polio had to be fought, people had to donate, everyone had to sacrifice. The other thing was that DNA was not an icon of identity, right? Jimmy Sarkett's or Sukkot's viruses, of course, we could talk about the ways that could re-identify, we can go back and find it, it could be labeled, but no one cared about cells. When blood cells were donated, they were donated, and no one thought, let's track down those blood cells, are they still me, are they still immortal, right? Because DNA was not thought of as an icon of identity, nor did the Lacks family think of it at that point as an icon of identity. Tuskegee, and I might add Guatemala, was still ongoing in 1947 and 1951 and still unrevealed. And up at that point, people still thought this was perfectly okay to have a differential between what doctors knew and what patients knew. Next. HeLa cells begin in that climate, and yet now we think of science differently. We think of science, and many of you colleagues think of it in some ways as also a source of profits. That's why it's called BioBank. That's why it's called material transfer agreements. That's why people at my university want scientists to develop things so they can make money. And this is not seen as a problem. It's just seen as different than it was in 1957 or 49. It's seen as part of a larger marketplace. Healthcare in 47 and 51 existed before Medicaid and Medicare. It rested on this earlier idea of beneficence. Care for the poor was a social obligation. Cold Spring Harbor, funded, of course, by Roosevelt, D.T. Watson's Place, were parts of charity for the poor. Next. But the lack of beneficence that was revealed in something like Tuskegee led to a larger lack of trust. States turned out to fail to protect the vulnerable. Individual doctors could not be fully trusted to act like Salk did. Hence, researchers are encouraged to use the tissues and cells to make products. And in part, it's how my university is funded. Lyrica, when I say more, which funds a drug that was developed at Northwestern, patented by Northwestern. Now part of the way that I'm in fact paid is through the profits by the Lyrica funds. And we think of that as the normative way. I'm sure Hopkins does the same thing for Dr. Sugarman. In part, you're funded by the good work that's done there and the way that the marketplace and universities cooperate in creating those profits. Next. But our sense of fair and unfair in medical research is somehow triggered by this case. The language of autonomy and the power of the marketplace did seem to us to be the best bet for persons, because now persons were seen not as patients but as consumers. Research subjects become routinely paid, and unlike polio trials where we didn't make a nickel for being, in the trial, it's expected now all over my university that there'll be some payment exchange for research participation. Cases like Moore and the Havasu Indians are based on this sense of unfairness that somehow emerged in this marketplace, and on this sense of the self being violated. Next. The core questions become, for an autonomy-based response like you heard Dr. Sugarman give, and where my whole field gives, who is the subject of rights? Whose rights are primary? How are the rights protected? What is the cost of bodies as things, right? And then who owns your body, which is the claim of the book and the claim of my field, bioethics. Next. But let's see if that's enough. Let's look at the 4 principles of bioethics: beneficence, non-maleficence, autonomy, and justice. Have they been enough? And the answer is empirically no. Could we see another sort of duty beyond the principles that have guided us so far and really are put at such risks, even with the best protections as Jeremy describes? Next. So I'm going to suggest that we should change our question. Rather than saying who's the subject of rights and then developing an elaborative normative structure to protect rights, we should ask different questions. What are our bodies made for? Is it possible that our bodies are made for the other, for one another? Who is the subject not of our rights, but who is the subject of our duty? What do we owe each other? And what's the duty of the medical researcher towards the vulnerable? Next. So when I think here about duty, then I have to ask the question, where does this duty come from? And for many Americans, one source of this duty is a religious source, a biblical source. For Jewish and Christian and Islamic thinkers in ethics, there are some suggestions of answers, and because I speak from Jewish traditions, let me give you a few that come from my tradition. There is, in first, a duty to heal that comes from the fact that the world itself is unfinished. The fact that illness and suffering exist in the world calls out to finish the world and respond to suffering. And the second duty that emerges from these texts is the duty never to stand idly by the blood of the neighbor, widely shared within all these biblical traditions. And I could give you a longer talk about Eastern religions and where they derive their duty from, but very similar calls. Never stand idly by the blood of the neighbor, because in some sense we are made for one another. Next. And there I'm going to suggest Fidelity, a new bioethics of duty, my new stem cell line here. The model for fidelity, as opposed to autonomy, is based on this duty-based understanding. And the model here is a debt relationship between a doctor, researcher, and patient, in which the doctor has a debt to the patient. Jeremy became a doctor because he learned on HeLa cells. The debt then he acquires, that he's used them, and he has to then pay back a debt. And that has a relationship of fidelity that's far deeper than the relationship of her autonomy. It's a much better way to describe the relationship. The promise here is justice, that the corners of the field belong to the vulnerable and the poor. The promise then is for witnessing and duty and altruism that everyone gets engaged in by developing transparent and honest work that's based not in the promise of profits, but in the courage to fail if the experiment fails. Next. The duty is transparent for all people, and there's 2 sorts of duties. For the researchers, is there a limit on the profits of your work? If so, what's that limit? Or is there a reasonable appeal for justice in your research? And what should that appeal be? Questions for the subjects. Is it possible to give freely without reward in a market-based society, even if it doesn't feel fair to you? Is that possible? Next. I'm going to end on a personal note. Like many of you in this audience, I struggle with raising a child with severe disabilities. And I want you to know that if I could cut off my left arm and hand it to Sean Morrison, who you're going to hear talk, and that arm would then cure people of that disease, my son, and prevent other families from facing what our family has faced and struggling to deal with his disease, and make it that nobody ever was faced with those sort of tragedies and dilemmas that we face on a daily basis. I would do it gladly. And I wouldn't care if Dr. Morrison became a gazillionaire if my left arm was able to cure my son. And I would warrant that every single parent out here would make the same claim. And I'll go further. I would warrant that Henrietta Lacks, if she was told that her children did not get polio and that millions of people were saved by her tumor cells, she wouldn't want a cent from Johns Hopkins. I would warrant that, given her background as a strong woman with deep religious beliefs, that she would applaud her gift to the world. And that is the basis for the new Fidelity. Next. Okay, but I'm a practical sort of girl. Fidelity means recognition must be given if it is wanted. Anonymous, fine, but recognition if it's wanted. It means your research must be transparent. And finally, researchers, why people distrust you is because lack of transparency and because this sense that the corners of the field are somehow not for the poor. I've defended stem cell research for the last decade. I think it's a good and right and just use of human imagination. And I think it warrants sacrifice for patients and for providers and for researchers, all of us. But I think if stem cell research is promulgated and makes all the wonderful progress, but the poor, like Henrietta Lacks' children, still don't have health care, it won't be a just science at all. So somehow, when we are developing it, we can't not only develop the rules about normative prohibitions, not only the rules of autonomy, but somehow to think about the rules for justice. The biobanks aren't banks. They're libraries. They belong to all of us. The research in some fundamental way belongs to all of us. Next. Thank you. For my colleagues at Northwestern University, for the parents, patients, and the advocates, for ISSC, our colleagues, and the GPI, and Bernie, for a decade of work on behalf of researchers and families. Thanks.
I'd like to thank Bernie and Alan for the opportunity to be here today. I'm incredibly honored to be a speaker on the panel and to bring a patient advocate perspective to the discussion on stem cells. I have Henrietta Lacks to thank today as well. From what I understand, many years ago, a scientist accidentally poured a chemical on a HeLa cell that spread out its tangled chromosomes. And scientists then use this technique to determine that humans have 46 chromosomes. And without this discovery, it's unlikely that several types of genetic diseases, such as Niemann-Pick, type C, would even be discovered today. I don't have slides today. I'm just going to tell you a personal story. It's a story that's transpired over the last 3 years, and it's a story about my twins, Addi and Cassi, and my journey to essentially save them from a dreadful disease. As Ed mentioned, 3 years ago, my husband and I received the worst news that parents could receive. Our twins were diagnosed with this genetic cholesterol condition, and it's called Niemann-Pick, and it's a progressive neurological condition that involves cholesterol storage, and it's fatal normally in childhood. The condition is autosomal recessive, and my husband and I both passed a genetic defect on chromosome 18 to our twins when they were conceived. And when we learned of this, there were no approved treatments for the condition, and honestly, we were told there was nothing we really could do, and there was really little hope. Prior to the diagnosis, I must admit that I had never really come very close to science, and as close as I came to science were political science courses in college, like the governor mentioned earlier. I never would have dreamed I'd be standing here today in front of stem cell researchers and industry to talk about the future of stem cells. Soon after my twins were diagnosed, I was really trying to wrap my arms around this nightmare, and I had a discussion with a neurologist at the Mayo Clinic. This is our twins' neurologist, and I asked him what the biggest hurdles were in Niemann-Pick disease. And besides only having 500 patients worldwide and very limited funding that mainly comes from parents, what was really setting us back from accelerating research? And what he told me was that we really only had a fibroblast model for testing, and we didn't have a neuron model. And despite Niemann-Pick being a lysosomal storage disease, that ultimately it was a neurological condition. And I remember at the time thinking, when we were talking on the phone, a neuron model, what the heck is that? I went on the internet and I started researching everything I could, and how to make neurons, who could make neurons. I'm sure this sounds pretty crazy, but at the time, that was my reality. I started reading about the potential of iPS cells and how these cells could be made from a person's own skin. And so I started contacting various researchers, some at UC San Diego, which in fact I'm now funding some research with Larry Goldstein's group there. It's a long story about how I became connected with MIT's Whitehead Institute, and I don't have time to get into that part today, but essentially MIT was making Alzheimer's iPS cells, and there are a lot of parallels between Alzheimer's and Niemann-Pick. So they were convinced, as was I, that Niemann-Pick iPS cells could be important for science. One thing that I'd like to mention is with ultra-rare genetic diseases such as Niemann-Pick, these stem cells are really game changers. Most conditions, there's very little research, and all of a sudden you can, essentially start studying these genetic diseases in ways that you've never really looked at before. So over the past year or so, MIT has reprogrammed a number of Niemann-Pick cells, including the twins cells, into iPS cells, and they've also figured out a way to differentiate these cells into neurons. And researchers there also wanted to compare the genetic defects from my husband and I as we pass these gene defects to our twins, and they've actually reprogrammed us as well. So we quite possibly could be the first family reprogrammed in this manner. It's unclear, but in any event, twice in the last year I've flown cross-country to visit the MIT lab, and I guess I've seen my second home, so to speak, where my cells and my twin cells are living in these heated incubators in these little round Petri dishes, and they're essentially taken care of around the clock. I don't really know how to describe what it's like to see your children or yourself, reprogrammed under a microscope, but I can tell you that it brings me great hope. I don't know how the cells will be used entirely in the future. We have some idea of what we want to do with them in the near term, but as Laurie was mentioning, if somehow my family or our cells could be used to help solve the mysteries of cholesterol in the human body, even if it helped one person, it would be an honor to us. So I'd like to just spend a couple of minutes talking about what we're going to do with the neurons. In a few weeks, I'll be heading to the National Chemical Genomics Center with Dr. Chris Austin's group, and some folks from MIT will be there, and we're going to be talking about the idea of high-throughput screening techniques on these neurons. With the goal is to test every FDA-approved drug. Dr. Austin has a massive library of compounds, but what we're doing is we're trying to get our hands on any FDA-approved drugs that may help move cholesterol out of the neurons that could help not only my kids but other kids that suffer from this condition. MIT has also spun up a project where they're working to try to correct the genetic defects that are in the cells, and we're in a very fortunate position that our genetic defects have been identified. So we have a real advantage there, and so that they're looking to do that. Whether these alleles can be corrected or whether these neurons can someday be used therapeutically, it's unclear at this point. Also, I'm making a genetically engineered mouse right now of my twins' cholesterol defects. One mouse is of myself and one is of my husband. This is underway in Australia right now. And once these mice are made, we'll be breeding them to create the Addi and Cassi mouse. We're really pursuing a path of personalized medicine, and our idea is to do different types of stem cell experiments on this customized mouse. So if there's anyone out there that has cells that they think we might be able to try, I'd be very interested. And finally, I just wanted to talk about some of the HIPAA laws and these privacy laws. These are something that I really personally would like to opt out of. They're holding me back. They're holding our researchers back. There's just so much approval that needs to happen on human subjects. And these IRBs are just an absolute nightmare. And I don't understand, how we can try to do regenerative medicine, especially when we're using people's own iPS cells, if you don't even know who the patients are. I mean, this takes a lot of time to develop, and, if our goal is to try to put cells back into Addi and Cassi, it's really kind of a joint effort between scientists and the government and patient advocates, the patients themselves. So I think it's just a real issue with some of these laws, and also I think a lot more time needs to be spent in how to get clinical trials up and running and how to fast track this with the FDA, because this is a real issue. I have another issue that was mentioned earlier about the FDA that I've been working with over the last couple of years, and I just think that's going to be a huge hurdle, and that's something that I'm going to be working on, because I have confidence that we're going to get cells. And then the question is, once we have them, will the FDA allow us to move forward to try to save our twins' lives? So thank you very much.
So in less than 1 hour, you have heard a very detailed history of what has gone on. You've heard some ideas about how to approach this research in the future, and you've heard a personal approach that talks about, again, some of the obstacles, the research obstacles, the NIH, the IRB, the FDA kind of obstacles. We have to remember that what happened in 1950 was fairly common. Laurie points out that a lot of this research happened on the poor and disadvantaged. Actually, that is true, but it also happened sort of routinely. If you look at hospitals in the 1950s, it was fairly routine to have people with their buckets standing outside surgical suites saying, "Do you have any spare tissue?" And they didn't care whether it came from poor people, rich people. They just wanted the tissue. So we have to worry about discrimination, but it was a more pervasive kind of issue. You heard that we all agree consent is necessary, but consent is an empty term unless you really unpack it and think about it as an informed choice, where someone is informed and they are able to choose. Even more important and a harder question is the question that all our panelists have brought up, which is, how do you keep the subject informed about not just what you're going to do today, but what you might want to do tomorrow, what you might want to do in the future? We have these cells. They are, as Chris has so eloquently described, sitting in a freezer somewhere. And what if 5 years from now somebody says, well, I have an idea? So there's that informational question. You heard a little bit about the importance of the marketplace, and that is certainly something that academia has to worry about now when we worry about funding issues. But even without the marketplace, most of the scientists that I know and that all of us know aren't really in it for the money. They're in it for the science, and they're looking at what can they do, what can they develop. Finally, what we want to leave you with is this notion of community, this notion of it takes a village to do a research project. The idea of, as Laurie puts it, the fidelity debt relationship. If you listen to what Chris is trying to tell you, here is someone who has children with an ultra-rare disease, and she's saying, I'm willing to share whatever you need if you're willing to make a deal with me that you'll focus on my needs. And that's really what we're talking about in science. If my hypothesis, if somebody had said to Henrietta Lacks and her family, I want your cells not because I'm trying to create something for my own self-aggrandizement, but because I'm trying to create something that will help me understand cancer, I agree. The answer probably would have been, sure, I don't want them. You can take them. But it is that kind of trust relationship that we are trying to create in a research environment. So I'd like to thank the panel for a terrific discussion. And thank you all for listening.